I want to thank the organizers for inviting me to present this work to you today. Alert to physicians in the United States. The statutory exemption of DHEA from clinical trials may be exacerbating COVID-19 morbidity and mortality. The United States leads the world in the number of COVID-19 deaths per 100,000 population. This begs the question, what is different about the United States compared to these other developed nations that is driving this disparity? There are, of course, many potentially contributing factors. Today I'm going to make the case that the unrestricted over-the-counter availability of an androgen, dehydroepiandrosterone, DHEA, uniquely in this country is contributing to the excess COVID-19 mortality that we're seeing here. It is well established that COVID-19 is an androgen-related disease. Males are affected more severely by COVID-19 than females. And androgens upregulate the ACE2 receptor to which SARS-CoV-2 binds to gain entry into the cell, as well as TMPRSS2, the transmembrane protease that primes the viral spike protein for efficient infection. DHA is the base androgen from which all others are synthesized, and its circulating form is DHA sulfate, DHAS. Primates evolved a unique androgen system in which the adrenogonadal primordium differentiates into an adrenal gland capable of synthesizing and secreting enormous amounts of DHA sulfate. This distinguishes primates from all other lineages, and humans are distinguished from all other primates by having by far the highest levels of circulating DHA sulfate. In humans, adrenarche, the onset of synthesis and secretion of DHA sulfate, occurs at about age 8 with the maturation of the adrenal zona reticularis. In humans, but not chimpanzees or other higher primates, adrenarche occurs as a developmental period completely separate from gonadarche. The human androgen system is characterized by extremely high blood concentrations of DHA sulfate and extremely low blood concentrations of DHEA. The ratio of circulating DHEA molecules to DHA sulfate molecules is roughly 1 to 10,000. As we shall discuss, the reason for this difference is that while DHA sulfate is virtually non-toxic, DHA is very different, capable of initiating a catastrophic surge in reactive oxygen species, ROS, and reactive nitrogen species, RNS, with lethal consequences to the cell. The adrenal glands of mice and rats cannot produce DHA sulfate, and consequently their circulating levels of this androgen are 10,000-fold lower than are found in humans. DHA sulfate is hydrophilic and requires transport by organic anion transport protein, OATP, to cross the cell membrane and gain entrance into the cell. Once in the cell, DHA sulfate can be desulfated to DHEA. DHEA, in turn, serves as the precursor to all androgens. This slide shows that induction of ACE2 and TMPRSS2 are induced by androgens, assisting in the infection of cells by coronavirus. The enzyme glucose-6-phosphate dehydrogenase, G6PD, is the source of most intracellular NADPH. NADPH oxidase, NOx, is one of the principal defense mechanisms against coronavirus and other viral pathogens. This was important to note because DHEA is a potent inhibitor of G6PD. Inhibition of G6PD activity sensitizes cells to coronavirus infection. There is evidence that this is mediated by inhibition of NADPH oxidase activity. So as you can see from this slide, DHEA can synergize coronavirus infection by at least 3 different mechanisms. The induction of ACE2 cell surface receptor, to which SARS-CoV-2 virus binds, the induction of the TMPRSS2 protease that primes the SARS-CoV-2 spike protein for efficient infection, and also by depleting the intracellular NADPH required for NOx activity. Here are some of the references showing that inhibition of G6PD activity enhances SARS-CoV-2 infectivity. DHA is regulated as a controlled substance in every developed nation except the United States. In the United States, DHA is available over the counter without any restriction whatsoever and without any clinical trials demonstrating safety ever having been done. How did this happen? The simple answer is that the United States has a very powerful supplements lobby, and it was able to pass the Supplement Health and Education Act of 1994. Prior to 1994, DHA was regulated in the United States as a controlled substance, as it was in every other country. But the 1994 Act exempted DHA from clinical trials to show safety. This exemption was based upon an assumption of safety, literature reports suggesting lack of toxicity in rodents. But humans have evolved a completely different androgen biology than have mice and rats, as we have already mentioned. An adrenal gland specialized in the synthesis and secretion of DHA sulfate. As a result of this 1994 law, DHEA was reclassified as a dietary supplement. This allowed it to be sold without any clinical trials demonstrating safety, sold in any amount, and used without any form of physician supervision. This is in regulatory antithesis to DHA's status as a controlled substance in every other developed nation. Oral DHA is marketed to aging males in this country as a longevity panacea. This is exactly the population that most frequently suffers poor COVID-19 outcomes. This is a screenshot of an advertisement for DHA on the web that I took last week. The medicine cabinets of aging American men have been flooded with cheap, high-potency formulations of oral DHEA. Ingesting a single 100 milligram capsule of DHEA, such as those shown here, is sufficient to elevate blood concentrations of DHEA into the supraphysiological range for most men. Because it is lipophilic, DHEA freely crosses cell membranes. Oral DHEA thus bypasses all of the normal safety mechanisms that have evolved to prevent it from reaching high intracellular concentrations. All these problems are magnified by the type of enzyme inhibition that evolved for DHEA, which I have marked here with a star. DHEA is what is known as an uncompetitive inhibitor of G6PD. Because uncompetitive inhibition can easily become irreversible, it's virtually unknown in natural systems except for DHEA. The reason that it can become irreversible has to do with the binding site of the inhibitor. In uncompetitive inhibition, no binding site for the inhibitor even exists until after the substrate binds. Substrate binding causes the enzyme to flex, and that flexing changes its conformation and creates the binding site for the inhibitor. As with other forms of enzyme inhibition, Unmetabolized substrate builds up in uncompetitive inhibition, but unlike other forms of inhibition, this buildup of substrate drives uncompetitive inhibition to extremes because more G6PD molecules have their DHA binding site opened up. Uncompetitive inhibition thus rapidly becomes irreversible. I often refer to DHA sulfate and DHEA as the Dr. Jekyll and Mr. Hyde of androgen biology. DHA sulfate is an ineffective G6PD inhibitor, so it can safely circulate at extremely high concentrations. And as we mentioned before, entry into the cell requires facilitated transport. In other words, it waits for an invitation to enter the cell. DHA is very different. It's a very powerful and competitive G6PD inhibitor. That's why it has to be kept at such extremely low blood concentrations, 5 orders of magnitude less than for DHA sulfate. Because it's lipophilic, it freely passes into cells, so it basically enters without an invitation. Renowned British enzymologist Athelstan Cornish-Baldwin put it this way: any metabolic pathway in which uncompetitive inhibition can occur can potentially respond catastrophically to the presence of inhibitor. So this begs the question, why would humans have evolved such astonishing levels of DHA sulfate when it is one desulfation step removed from the irreversible, uncompetitive inhibition of so critical an enzyme as G6PD? My lab has recently been able to answer that question. The answer is exactly as Cornish-Baldwin suggested. The human DHA sulfate, DHEA androgen system evolved to inflict catastrophe on particular cells, cells that we call singularities. We define singularities as cells in which the p53 tumor suppressor gene has been inactivated. My lab has gathered data supporting that DHA sulfate, DHEA are the core elements of a human-specific kill switch tumor suppression system designed to extinguish singularities while they still exist as single cells. Here are some of the references related to that work. And now I'm going to show you how this kill switch works and why it is related to the COVID-19 pandemic. G6PD can be thought of as the NADPH factory of the cell. My lab showed that G6PD activity regulates the mevalonate pathway because of that pathway's extreme sensitivity to intracellular NADPH concentrations. The NADPH produced by G6PD is required for the synthesis of the antioxidant selenoproteins that prevent reactive oxygen and reactive nitrogen species from overwhelming the cell. NADPH is also necessary for maintaining antioxidant selenoproteins in their reduced state, and to maintain ubiquinone in its reduced state as ubiquinol, necessary to prevent cell death by the process of ferroptosis. In this cartoon, antioxidant selenoproteins are depicted as the firemen of the cell, and their fire hydrants are filled with flame-retarding NADPH. As shown in this slide, in the normal cell with active p53, DHA sulfate stays in the blood. In singularities, organic anion transport protein, OATP, and the steroid desulfatase, STS, are activated by p53 inactivation. This results in DHA sulfate being imported into the cell and desulfated to DHEA. DHEA then blocks NADPH synthesis by its irreversible uncompetitive inhibition of G6PD. The selenoprotein firemen of the cell disappear and their fire hydrants run dry. This results in the destruction of the singularity while it is still in its single-cell state before it can evolve into a tumor. Because it can pass right through cell membranes, oral DHEA bypasses all of the controls that have evolved to prevent kill switch activation in normal, healthy cells. How is this related to the COVID-19 pandemic? Infection with SARS-CoV-2 is associated with systemic inflammatory reactions, not only in the lung, but also in the digestive tract, kidney, cardiovascular system, brain, liver, and skin. Such extrapulmonary inflammation dramatically increases COVID-19 morbidity and mortality. It's quite clear now that this COVID-19 related systemic inflammation is caused by endothelialitis. Due to the exemption granted by the 1994 Supplement Act, the effects of ingestion of oral DHA have not been studied in humans. But in dogs, my laboratory has shown that it produces a systemic inflammatory reaction that is focused on the vascular endothelium. This tissue specificity is alarming. By inducing the kill switch in normal endothelial cells, oral DHA may precipitate or exacerbate the endothelialitis underlying systemic inflammation observed in COVID patients. In our canine work, susceptibility to the induction of systemic inflammatory reactions by DHEA appeared to be due to two major mechanisms. The first of these mechanisms is by the uncoupling of endothelial nitric oxide synthetase. Thus, G6PD provides the NADPH required for the synthesis of tetrahydrobiopterin, BH4, and that is required for enos to function properly and perform its job of creating nitric oxide. Now, in the DHEA-treated cell, where DHA inhibits G6PD, NADPH disappears. And when that happens, enos becomes uncoupled, and instead of producing nitric oxide, it produces peroxynitrate, a very potent reactive nitrogen species. So this reactive nitrogen species is dramatically elevated in endothelial cells exposed to DHA at the same time that DHA has caused the disappearance of the selenoproteins required for the detoxification of peroxynitrate. Another reason why DHEA toxicity is most pronounced in endothelial cells is that DHA inhibits NADPH oxidase in a tissue-specific manner as well. So again, G6PD produces the NADPH that NOx requires to inactivate coronavirus and prevent infection. When DHA inhibits G6PD, NADPH become depleted, NOx cannot function, and coronaviruses have free rein to infect. References for all of the above can be found in my recent reviews cited here. Let me conclude with a brief summary of all of this. The Supplement Health and Education Act of 1994 exempted DHA from clinical trials based upon the absence of literature reports of toxicity in rodents. But humans evolved an androgen biology highly sensitive to the ratio of circulating DHA sulfate to DHEA. Mice and rats evolved no such system, so toxicity data in them is meaningless. Oral DHA bypasses all of the controls that evolved in humans to prevent irreversible, uncompetitive inhibition of G6PD from occurring in healthy, normal cells. Because of the exemption granted to it by the Supplement Act of 1994, no clinical trials demonstrating safety in humans, particularly SARS-CoV-2 infected humans, have been performed. In the absence of such clinical trials demonstrating safety, the unrestricted over-the-counter availability of DHEA during the current pandemic is not a rational approach to public health. There is a high likelihood that oral DHEA, uniquely available in this country as a dietary supplement, is responsible for a significant fraction of the excess mortality from COVID-19 observed in this country. Thank you for your attention.